In a perfect world, you would have the same protein encoded for by the same RNA encoded for by the same DNA every single time. But there are going to be problems. In this lecture, we're going to identify different types of mutations and how they might affect the protein's structure and function. Translational errors occur when there's a mutation on the DNA. There are three types of mutations: additions, deletions, or substitutions. An addition is where a new nucleotide is added. A deletion is where one's taken away. And a substitution is where you swap one for the other. Which of these three mutations is going to be the most serious? I'm sorry, the least serious? Substitutions. Most single nucleotide polymorphisms, one nucleotide changed, that we survive, the non-lethal ones, are termed substitutions. You're just swapping one out, because typically they're only going to affect a single amino acid. Yes, this will affect the conformation of the protein product, but that protein may still be partially functional, which means it's not going to kill you. There are certain genes that if you change one nucleotide in, you're going to die from that. It's a mutation. If, however, you added a nucleotide or deleted a nucleotide, then you're going to have what's called an addition or deletion, a frame shift mutation. You've shifted the reading frame. So you're not just changing one amino acid, you're changing every amino acid after that mutation, which will definitely change the shape and structure of the polypeptide. Your protein will no longer have the same form, it will no longer have the same function. A lot of times these break down quickly and you don't survive this one. The most talked about human condition caused by single nucleotide polymorphism is sickle cell anemia. Sickle cell anemia is caused by one amino acid change, one substitution, in one spot, in one protein. Yet, it will change the shape of your blood cell, every blood cell in your body, so that they're sickle-shaped. They will cling together more. They'll lead to clots, and that could possibly kill you. So one change in one amino acid can cause this condition. This single nucleotide polymorphism will cause sickle cell. Or mutations may cause no change. Here, we have TTC at the DNA level. That produces lysine. Turns out, though, if we were to change that C to a T, TTT is also lysine. No change. Yeah, there's a mutation, but it's a silent mutation. You can't see it. It doesn't do anything. Some obviously will have bigger impacts. ATC, so if you change the first T to an A, so instead of TTC is lysine, ATC is stop, that will have a big difference in the shape of the protein, because suddenly there's a premature stop signal. So mutations will happen, permanent mutations happen on the DNA, and they could be additions, deletions, or substitutions. Substitutions are usually not as serious as additions or deletions, because additions and deletions are both frame shift mutations. They shift the entire reading frame. So this was a short but important lecture. Translation errors. These are mistakes or inaccuracies that occur during the process of protein synthesis. And they can happen. We went through all these different processes on how we can stop them from happening, but they still can occur. When they occur, they might lead to non-functional or malfunctional proteins that don't work as well as they otherwise would have. We talked a little about missense mutations, errors that result in when you substitute one amino acid for another. That changes the protein's function. One slight amino acid change may not have a big impact, but it can still change the function of this large polypeptide. There are nonsense mutations. These are basically nonsense mutations are where a change in the amino acid sequence or of the RNA will lead to a stop codon occurring before the protein was actually supposed to stop. So you get a half-sized or part-sized polypeptide, and it doesn't have much of a function. And then you might have a frame shift mutation where you could insert or remove a base, and it changes the reading frame of the ribosome so that you're not getting the right amino acids. After that frame shift mutation, after that addition or deletion, every single amino acid after it's affected. So you get an entirely new polypeptide chain. More often than not, these are deleterious and they hurt the organism's fitness. These translation errors are caused by mutations in DNA sequences. Mutations in things like RNA won't have a long-term effect. Mutations in DNA are permanent. They stick there. It's a mutation in the regular or the main the copy, the master copy. So with DNA errors, with translation errors, you can in fact lead to serious health conditions. These content review questions are here to focus your studies. In the next set of lectures, we're going to be going over genetic regulation. So we talked about how we can build proteins from DNA. Now we're going to talk about how we can control how many proteins are being produced at a time, or how we can produce proteins only when we need them. So that's coming up in the next set of lectures.